So here in this second video of Respiratory System Part 1, we're going to extend our discussion regarding ventilation into breathing mechanics. And when we think about breathing, and this is just reiterating what we had discussed in the first video, breathing, the sole purpose is to move air into and out of the lungs so that we can use that air to participate in gas exchange. And just as a reminder, recall that ventilation is the act of moving air, whereas respiration, you need to think about the exchange of molecules between two different physiological systems. And so with external respiration, we're exchanging molecules between the respiratory system and the cardiovascular system at the level of the alveoli. Internal respiration is the same thing, with the exception that we're talking about the cardiovascular system and tissues throughout the body. So what's important to take from this one particular slide is that ventilation leads to respiration. Without ventilation, we would not be able to engage in gas exchange. And so ventilation exists in two general phases, and we'll take a look at these in more detail in a later video. But those two general phases are inspiration and expiration. So when we talk about inspiration, air is flowing into the lungs, and in this lecture we'll talk about the mechanisms that we use to push that air, or to pull that air into the lungs, as well as pushing the air out of the lungs with expiration. And this is all driven by pressures, and we'll talk about Boyle's law here on the next couple slides. But in order to manipulate the pressure, we have to change volume. So by changing volume, that will have a direct effect on pressure changes, and it's the pressure changes that are going to drive ventilation as well as respiration. And we'll get into that much later as well. But when we talk about breathing, recall that breathing is just airflow, and it's a mechanical process, which means that we have to change the dimensions of the thoracic cavity, and by changing the dimensions of the thoracic cavity, we're changing the volume, which is going to lead to a change in pressure. So volume changes will lead to pressure changes. And it's the pressure changes that will drive the flow of these what are called blood gases, oxygen, CO2, to participate in respiration. But coming back to Boyle's law just simply states that there is an inverse relationship between the pressure of a gas in a container and the volume of that container. So we're going to take this very simple idea and apply it using a very simple geometric representation to that of the lungs and the alveoli. So here we have a container, standard a cylinder, and inside the cylinder we have a number of molecules. Now those molecules are in constant motion, which means that being in constant motion, those molecules will impact and hit the sides, the internal surface area of that container, and apply a force. And so whenever we have an impact, we have a force exerted by these molecules on the interior of this container. Now if we take the same container and we stretch it out, doesn't matter which direction, but if we stretch it out and increase the internal surface area without adding any more molecules or losing molecules, so we have the same number of molecules in there, those molecules will still collectively hit the sides of the container, but because it'll take a longer time to hit the container, the overall pressure will decrease. And so the impact of one molecule against the container, as we can see here, will exert the same force as it exerted here, except that we'll have a lower number of collisions along the interior of that container. So what we clearly see moving from the left to the right is that when we increase the volume, we increase the interior surface area, and we reduce the number of collisions between those molecules and the container itself. And so the particle force is the same. So if we focus on the one particle, it's going to exert the same force against this wall as it did prior to the expansion of the container, but the pressure will be much lower, and it's driven by this simple equation that the amount of force, despite the force being the same, if we increase the denominator, which is the internal surface area, the quotient, the pressure will be much lower. And it's that pressure in the lungs that's going to drive the movement of air, whether the air is going to leave the lungs or whether we're going to pull air into the lungs. The next two slides presents a table that's essentially a summary of the mechanism involved with inspiration and expiration. I'm going to skip through slides number 4 and 5 fairly quickly because I'm going to talk about these mechanisms in a little bit more detail. But no, slides 4 and 5 are two very good slides to come back to after this lecture is over to capture that summary of the two basic stages or phases of breathing, which is inspiration. And so recall, using Boyle's law, that when we talk about inspiration, we have to change the pressure so that it's going to pull air into the lungs, and expiration, we have to change the pressure so it's going to push air out of the lungs. And let's take a look at how we do that. So if we start on the left-hand side, here we see this very simple schematic picture of the diaphragm muscle. And so the diaphragm muscle is our main inspiratory muscle. And in this blue here, we have a depiction of a single alveolus, the bronchiole, bronchus, and trachea representation. And if we take PB, which is the barometric pressure, that's the pressure exerted by all the molecules in the air outside of our body. If we set that relatively to a relative value of 0, we know in reality the absolute value is not 0. It's on average 760 millimeters of mercury, but for our illustration here, we're going to set it to 0 and use that value to compare to the pressure changes that take place inside the lungs. So just imagine that you've just finished pushing air out of the lungs, so the end of expiration, and there's a small period of time in which you're not ventilating. There's a very small period, short period, where there's no air movement. And when there's no air movement, we can assume that the alveolar pressure, the pressure exerted by the air molecules inside the alveolus, is going to be 0. Barometric pressure is 0. If the pressures are equal, we don't have airflow. So there's no air movement in that very short period of time at the end of expiration. Moving over to the right-hand side, we still have our barometric pressure of 0, but the difference here is that we have the diaphragm muscle now contracts. And so by contracting, the diaphragm muscle actually moves in an inferior direction. Compare that to the left-hand side, where the diaphragm muscle appears a little bit more elevated, but when it contracts, it flattens out and it pushes in a downward direction, and by doing so, it increases the size, or it increases the volume of the thoracic cavity. So we have more room in here. But by expanding, by pulling on the lung tissue, which is attached to the diaphragm, we see an expansion of the alveolus. And so what this creates essentially is a vacuum. So the alveolar pressure now goes to a value of -1 with respect to the barometric pressure. And so because pressures go from high to low, 0 is obviously a higher number than -1, that vacuum is going to pull the air from outside the body towards the interior of the alveolus, and that's what defines inspiration. So inspiration is defined when barometric pressure becomes greater than alveolar pressure. Barometric pressure in and of itself doesn't change. It's the alveolar pressure that is decreasing because of the increasing volume of the lung tissue triggered by diaphragm contraction. So in this case, air moves in, so we have inspiration. Now here on slide 7, we're going to start on the left-hand side, and so here we have our barometric pressure. It's equal to 0. And at the very end of the inspiratory phase, so you've just taken in some air, the alveolar pressure has now climbed up from -1 to 0. Although we have the expansion of the volume of the alveolus, the pressure goes up a little bit because of the presence now of all those extra molecules that we took in during the inspiratory phase. And when we have that slight increase in the internal pressure of the alveolus, the alveolar pressure, that will equal the barometric pressure, short period of time, and we don't have air movement. We don't have a net movement of air. So everything stays where it is. And one more point to make before we move to the right-hand side. Here we see the diaphragm is still in a contracted state. But when we relax the diaphragm, so it's pretty subtle, but if you take a look at the illustration of the diaphragm muscle on the left-hand side and the right-hand side, you see the diaphragm muscle is in a more elevated position, and that's the position it takes when it's relaxed. But when the diaphragm reverts back to its resting state, it pushes superior on the lungs, and when it pushes superior, it decreases that thoracic cavity volume and the volume of the lungs. So with diaphragm relaxation, that decreasing volume will drive an increase in the internal pressure on the alveolus. So it moves from 0 to 1. So alveolar volume will decrease, intra-alveolar pressure increases, we keep our barometric pressure at 0, and this is what defines the expiratory phase. And that's when the alveolar pressure climbs above barometric pressure and air moves out. So notice at this point we haven't talked about gas exchange yet, O2, CO2, but we can't have gas exchange unless we have those ventilatory dynamics at play. So ventilation just doesn't happen on its own. We need muscular action for that to happen. And so we have this dome-shaped muscle called the diaphragm, as it's illustrated here in this picture inside the rib cage. And it's a dome-shaped muscle that has connections to not just the lungs, but also the heart. And the dotted line here illustrates the diaphragm in a relaxed state. But when that muscle contracts, it flattens out, decreasing the volume of the abdominal cavity while increasing the volume of the thoracic cavity. Because the diaphragm has connections to the spine and to the rib cage, this contraction will actually separate the ribs. We can't have an increase in lung volume without rib separation. And it's going to be even more difficult if we don't relax the abdominal muscles. So when we take a breath in, the diaphragm flattens out, the abdomen actually distends slightly to accommodate that diaphragm moving inferior. So during quiet breathing, typically the diaphragm muscle is the main one that's active, but in some cases the external intercostal muscles may assist the diaphragm muscle. And as you can see, that muscle, those muscles that are located in between the rib cages, and when these muscles contract, they have the effect of separating the ribs from each other. Now, when we exercise or we have a need to increase ventilation, increasing the depth of breathing, increasing the rate of breathing, we then activate additional muscles that we'll detail here on the next slide. And these additional muscles will cause a further increase in thoracic volume by creating more rib separation. So now in terms of expiration, under resting conditions, so when you're sleeping, typically expiration is a passive process. So the simple act of relaxing the diaphragm will resume its resting state by pushing on the lungs, and that is sufficient to decrease the volume, increase the pressure, and push that air out of the airways. But on occasion, we do need some additional assistance with pushing air out of the lungs when there's an increased need to ventilate. And so contraction of the abdominal muscles will move those muscles towards the interior of the abdominal cavity, and that'll decrease the volume, helping to push the diaphragm muscle into its relaxed state. And the internal intercostal muscles, as opposed to the external intercostal muscles, the internal intercostals are assistive with expiration. So these are two major muscles of expiration that are not necessarily active all the time, but when we have a need to increase ventilation, we start to recruit those muscles more. Now, just to review the muscles of inspiration, of course, the major inspiratory muscle is the diaphragm. In some cases, under resting conditions, the external intercostals may be active, but their activation certainly increases when there's a need to ventilate more. But when we're exercising, we see additional muscles helping to play a role in separating the ribs from each other, and that's the pectoralis minor, the scalene muscles, and the sternocleidomastoid muscles will all function to help create that separation to increase the volume of the thoracic cavity, pull the pressure down, create a greater vacuum, pulling more air into the lungs. So just to review, the diaphragm muscle is considered to be the floor of the thorax, of the thoracic cavity, roof of the abdominal cavity. The diaphragm muscle has connections to the lungs as well as the heart, and we'll get to that in the cardiovascular unit. The diaphragm muscle, because it is this dome-shaped muscle, it does have a hole or a foramen that allows the aorta and the inferior vena cava to traverse from the heart to the inferior part of the body. And it's an inspiratory muscle. The external intercostals, the 11 pairs, function as an inspiratory muscle by increasing the spacing between the ribs, while the internal intercostal muscles function to depress the ribs, pull them closer together, and by pulling the ribs closer together, that decreases the volume of the thoracic cavity, driving up the pressure, helping to push air out of the lungs. The abdominal muscles will compress the abdominal cavity, assisting the superior movement or the superior push of the diaphragm to reduce the volume of the thoracic cavity. And so it functions as an assistive expiratory muscle. Now, muscles carry out the mechanical action, but what drives the muscles? The muscles are driven by neurons. And so the neurons that control breathing originate in what's called the pons area of the brainstem. So we have a cluster of neurons called the pontine respiratory group, and these neurons will send axons to innervate a group of neurons in the medulla oblongata, which is inferior to the pons. And one cluster of neurons are just simply referred to as the DRG, or the dorsal respiratory group. Another group that sits anterior or ventral to that is the ventral respiratory group. And the dorsal respiratory group is particularly important because if you follow the pathway here, you'll see that it innervates a nerve called the phrenic nerve. And the phrenic nerve, if we follow it down, innervates the diaphragm muscle, that when it activates the diaphragm muscle, it triggers its contraction, thereby causing the first phase of ventilation, which is inspiration. So the medullary respiratory center contains the important dorsal group, which stimulates the diaphragm muscle. The ventral group is activated when the need to increase ventilation, the need to increase the depth of the inspiratory phase goes up. And so the ventral group will stimulate the intercostal muscles, will also innervate and activate the abdominal muscles, which will drive expiration as well. So typically when you're sleeping during rest, the dorsal group is the main one that's active, and we just rely on elastic recoil and relaxation of the diaphragm to push air out of the lungs for expiration when we're resting. But when there's a need to increase ventilation, the ventral respiratory group becomes more active. And regulating these two clusters of neurons are the clusters of neurons superior to the medulla oblongata, and that's the pontine group, those cluster of neurons in the pons area of the brainstem. And they help to regulate breathing rhythm so that we have that coordinated action between inspiration and expiration. So the overall control of the mechanical function comes from the neurons, but then that begs the question is what controls the neurons? Well, the whole system is all designed around maintaining a certain amount of oxygen in arterial blood, and that's the blood that is often referred to as oxygenated blood. That's the blood that carries the important oxygen molecules to our aerobic tissues, as well as maintaining a certain carbon dioxide pressure. We can never be completely without carbon dioxide, but we can't have too much carbon dioxide. And so our system is so designed to regulate how much O2, how much CO2. If it goes up or down, then our ventilatory system will respond to reestablish those concentrations. And there are two mechanisms to control ventilation. The first mechanism here is what's called efferent output from the cerebral cortex. And we have motor neurons to our respiratory muscles, and these are neurons that are under voluntary control. So you can actually think about taking in a deep breath when you're just sitting and you're not moving. You can think about and actually force air out of your lungs. That mechanism comes from these motor neurons that originate in our motor cortex, in our cerebrum. But when we're sleeping, we don't have to think about breathing, and the mechanism to control those muscles, as we just discussed on slide 11, comes from the brainstem area, the regulation by the pons driving the DRG, the dorsal respiratory group, to contract the diaphragm muscle. Now, that's the neurocontrol. Going back to slide number 12, the first bullet item indicated that the whole system is designed to maintain O2, CO2 presence in the blood. And so we have to have a way to detect any changes in those levels. And so if we have a change in carbon dioxide levels, if we have an increase in carbon dioxide in our arterial blood, because it is a byproduct of metabolism, so if metabolic rate increases, then we're going to see an increase in carbon dioxide production. That will trigger an automatic and reflexive increase in the rate and the depth of respiration, or rather ventilation. Hypercapnia is a term that refers to a greater than normal amount of carbon dioxide, and this is typical when we have an increase in metabolism. Hypocapnia is a lower than normal amount of carbon dioxide. And something that was mentioned on the previous slide is that our system is so designed to maintain a certain amount of carbon dioxide coursing through our cardiovascular system. But to be able to detect those changes in carbon dioxide, as well as the pH, the acidity level of our fluids, we have what are called chemosensitive areas in that brainstem region, particularly the medulla oblongata. So the cells here are particularly sensitive to any changes in the acidity of the fluid outside of those cells, or any changes in the CO2 levels outside of those cells. Carotid bodies, these are small clusters of sensory neurons that are embedded inside our carotid arteries, and they're sensitive to a few different types of things. One such change is blood pH. If blood pH decreases slightly, and exercise will do that, blood becomes slightly more acidic. That will discharge these sensory receptors, these sensors, if you will, that will feed information back to the brainstem area and increase ventilatory drive. Oxygen is something that we have to maintain in certain concentrations in our arterial blood, and so not only do we have receptors, these specialized sensors to detect O2 presence in our arterial blood in the carotid arteries, but we also have those sensors embedded in the aorta, that first large blood vessel that leaves the left ventricle that transports oxygenated blood to all the tissues in the body. So these are chemoreceptors. These are sensory neurons, particularly designed to pick up any decrease in oxygen. And if there's a decrease in oxygen in that arterial blood, it will send information back to the respiratory center in the brainstem and increase the activation of our inspiratory muscles. And when we have that decrease, that is by definition hypoxia. And so hypoxia is a condition where we have a lower or a decrease in the oxygen levels below normal values. And there are many reasons why this would happen. One such example is if we climb to a high altitude. So in the extreme case is an expedition to Mount Everest, which is very highly elevated above sea level. Typically, when a person goes to that level, they experience a reflexive increase in heart rate, a reflexive increase in respiration rate, because they're experiencing what's called a hypobaric hypoxic condition. They're going to experience a decrease, okay, in the blood, in their oxygen levels in their arterial blood, because of a decrease, a less than normal amount of barometric pressure because of the altitude level. And so what our bodies are so designed to respond to that by increasing ventilation to get more air into the lungs and to provide that oxygen. This last slide, we're just going to go through some basic definitions here that we'll touch on in some later videos. But minute ventilation, as the name indicates, this is a term that defines how much air we move into and out of our respiratory system each minute. And it's defined by something called the tidal volume. And the tidal volume is the amount of air we move with one breath. On average, it's about 5.5 liters per breath. And we multiply that by the respiratory rate. On average, we take about 10 to 12 breaths per minute. But as I mentioned, we'll revisit this term in a later slide. So respiratory rate, also referred to as respiratory frequency, is the number of breaths taken per minute. Now, not all the air we ventilate the lungs with participates in respiration, and that's called anatomical dead space. And so we have air that always exists in our nasal cavity, our pharynx, our larynx, trachea, and all the air passageways. It's always present, and it's important that we maintain a certain amount of air in all those passageways so that we don't produce a vacuum and collapse those air passageways. But that air does not participate in gas exchange, and so that's why it's referred to as dead space. Doesn't mean it's useless, it just means that it does not participate in respiratory action. That's a little bit different than physiological dead space. Physiological dead space is the anatomical dead space plus any air in the alveoli, the sites of respiration, that doesn't engage in gas exchange. So just because we move all that oxygen into the alveoli, it doesn't mean that all of that oxygen gets exchanged with the cardiovascular system. Alveolar ventilation, a very important term, because this refers to the volume of air that's available for gas exchange per minute. So we can actually ventilate the lungs, but if our alveolar ventilation is low, then that's going to hurt our respiratory rate. Two last terms to close out this video: ventilation versus perfusion. So we've already talked about ventilating, that's airflow. The second phase of this, which is really important, is perfusion. So although we may be well ventilated, we have to have a cardiovascular system a healthy cardiovascular system that will be able to participate in that gas exchange. And that's what the perfusion term refers to. It refers to the extent to which the cardiovascular system interacts or is physically connected to the respiratory system. So this ends our second part of respiratory system part 1, breathing mechanics. And the next video in this series you should watch is cardiovascular system part 1, which will detail the functions of the blood. Then we'll go back to respiratory system part 3 after viewing respiratory system part 2. Then move on to the cardiovascular system part 1.